Hi, my name is Faith Boynton, and this presentation is for the class Bio111 Fundamentals of Cellular and Molecular Biology at Mid-Michigan Community College. This presentation is on biosensors, specifically the biosensors PDUSC and PDON. To begin this presentation, it is important to understand a little bit about the background of this experiment. First off, what are biosensors? Biosensors are devices in living organisms that detect presence of chemicals. In this experiment, the chemical is light. The biosensors in this experiment are PDUSC and PDON. PDUSC and PDON are biosensors whose DNA systems allow people to modify its gene expression with light. The presence of light or no light control what color the bacteria will show. PDON will turn a different color in light, and PDUSC will turn a different color in the dark. My hypothesis consisted of if I expose both the PDON and the PDUSC to the same amount of light and darkness all day, I think they both will relatively stay the same color because the PDUSC is only in the dark for half the day, same as PDON. Then the PDUSC and PDON will be only in light for half the day. Therefore, the biosensors PDUSC and PDON are equal in terms of being in their proper growing environment. For my experimental design, I set up my lab as stated in the biosensor lab procedure packet. So first off, I created the agar, I poured my plates, I let them cool until they were solid, and then I added sterile water to my bacteria, shook it up, and swapped my plates. And I sanitized my gloves, my area, and my occlusion loops. Once I got all the plates set, I labeled them according to their corresponding biosensors. So for the PDON bacteria, I labeled the plates with PDON, and same as the PDUSC, I labeled them with PDUSC. I set them both petri dishes in the light at 7 in the morning. At 7 at night, I moved the bacteria back to a pitch black room until it was 7 in the morning. I did this rotation for 5 consecutive days. So in the morning at 7 a.m., I would put my bacteria in an open window with the curtains open and all the lights on in the room. At nighttime at 7, I would close the curtains in the room, shut off all the lights, and I would close the door so the room was completely black. My results consisted of my PDON plate containing about 6 colonies of bacteria, which were pink. They were almost as comparable to the color of flesh or salmon. On the other hand, my PDUSC plate only had about 6 colonies of bacteria that was not as pink as the PDON, almost white with a little bit of pink. Overall, my hypothesis is not really supported because initially I thought the bacteria would be similar in color, but the PDON was a lot brighter in color than anticipated. I was not surprised by the PDUSC actually, because typically the PDUSC is a lot lighter out of the two biosensors. The PDUSC is lighter, but I assume the PDON to be lighter or closer in color to the PDUSC. As seen below on this slide, my PDON and my PDUSC bubbles are pretty similar in the color of my petri dishes on the slide beforehand, and the PDON is very, very pink, and my PDUSC is almost white. If I was to reconduct my experiment differently, I think I could go down multiple different routes to do it. I would maybe start by switching the plates of bacteria in and out of light more frequently, maybe every 5 hours instead of every 12 hours. A few other things that I could also do would be just expose the bacteria to regular light, or I can put them in rooms where they'll just get regular daylight or darkness from the sky itself. Either way, I think the outcomes of them would be sort of similar to what my experiment is now.